Hi, folks. Thanks very much for inviting me to this summer school. It's wonderful to be here in Croatia. So again, to set the scene a bit, although I've ended up working in machine learning research, that was not my original background. I actually started out in computational biology, and I did that PhD at Oxford on simulating the reproductive system of a tiny millimeter-long worm called C. elegans. And after deciding that my PhD topic wasn't exactly going to set the world on fire, I actually left academia for a while and got a job as an honest-to-God software engineer at an online supermarket developing AWS microservices. And it was back in 2018 that I first heard about a job going at this London-based research institute called DeepMind. At the time, they were best known for games research. So in 2016, they developed the famous Go playing agent, AlphaGo. But now they were recruiting for engineers with a science background. And apparently the CEO's true passion was using AI to advance science. And he seemed to think that time was now ripe to push on that. At first, there were just two established projects and really a handful of people in the new science unit. But since then, it's grown to cover topics as diverse as weather forecasting, maths, fusion, and genomics. Now, for most of my time at DeepMind, I've worked on AlphaFold, and this is probably still the project that our science team is best known for, and it's definitely the story I'm always expected to tell in talks, so don't worry, I am going to cover it today. And essentially, AlphaFold is a network that tries to predict a protein's 3D structure from its sequence. And for this work, John Jumper and Demis Hassabis were awarded a share of the Chemistry Nobel Prize last year, together with David Baker. But an awful lot has happened in AI since then, in particular, the large language model revolution. And my more recent work has actually been in that space, investigating the ways that LLMs can best be applied in order to advance science. So in an attempt to try and tie these different projects together, today I'm going to talk about some of the work that I've been involved in at Google DeepMind, but also in the context of the wider AI for science environment. I'll discuss a bit how our ways of working there have shifted since 2016, and I'll talk a little about what it's like to do AI for science now in the age of Gemini and ChatGPT and all the rest. Small disclaimer on that, this is not an attempt at like a rigorous historical review of AI for science. It's mostly just a framing device to give the talk a bit of a theme, and everything that I'm going to say is obviously colored by my own experiences. So first of all, let's talk a little bit about the mid-2010s and about a project that happened at around that time, the development of AlphaFold 1. So I'm dating myself quite precisely here, but I was a student between 2008 and 2016, and it was very possible to go through your entire university career in computational biology at that time, bachelor's, master's, and PhD, without hearing about machine learning once. It was a very different time. And there also seemed to be this fairly long delay between new methods being reported in the ML literature and then actually finding their way into scientific applications. In fact, in the early AlphaFold team, we were actively discouraged from discussing ML for biology papers in the journal clubs for fear that the methods in them might be a bit out of date. So it was considered better to discuss either cutting-edge ML or cutting-edge biology, but not try and look at the intersection between the two. In general, the vibe at that time seemed to me to be that you can get a long way in ML for science just by taking well-established techniques and then minimally adapting them to fit your problem. And as we'll see, work at DeepMind also sometimes fit that pattern. So already in 2016, DeepMind had an established team working on protein folding. Now, proteins are these very important molecular building blocks in biology that perform all kinds of functions in the cell. They can be structural components in things like skin and muscle. They act as receptors and messengers for cell signaling, and they're key components of the immune system, to give just a few examples. And structurally, a protein is a 1D chain made up of about 20 different types of amino acid building blocks, kind of like beads on a string. that 1D chain spontaneously folds up into a complex 3D structure. And the process is driven by physical laws, by the interactions of amino acids with each other, and also by their properties and the interaction with the environment. Now, knowing the 3D structure of a protein is something that can be extremely useful. It can tell us something about how the protein works, because structure dictates function to some extent. It can tell us what other proteins it might interact with, and that's really important in biology. Almost nothing operates in isolation. Everything is part of this complex network. And it can even tell us how we might be able to modify a protein's behavior with small molecule drugs. There's the whole area of, structure-based drug development, where you're trying to tailor the shape of a molecule to fit into a particular binding site on the protein's 3D structure. So there's lots of interest in knowing 3D structures, but they are tough to figure out experimentally. The leading methods are still X-ray crystallography and cryo-EM, and even today, both of these approaches can run into intractable problems. I've had examples that I was emailed about during the AlphaFold process, where a scientist would say, we've been trying for 10 years to just make enough of this protein to be able to crystallize it. So you can fall really at the first hurdle. And as a result, protein structures can still take months or years to determine in the lab. By contrast, that 1D sequence is very easy and very cheap to work out. You can get it just by sequencing the encoding gene. So this was the research challenge in a nutshell. Can we predict a protein's 3D structure from its 1D sequence using a neural network? And if this were possible, we would then be generating valuable structural information from cheap sequence information, and we could save a lot of biologists a lot of time. Now, as I mentioned, a popular strategy at the time seemed to be to take an off-the-shelf method and then try to minimally modify it to make it fit your scientific problem. And DeepMind's very earliest attempts at protein folding probably fell into this category. There was actually a protein folding computer game floating around at the time a few years ago called Fold It, which was something that used realistic physics calculations in the background to try and work out, the energy associated with a particular protein configuration. And the challenge was for human players to try and fold up a protein to put it in the minimal energy configuration. So energy was your score in this game. And this project basically started out as a hackathon at DeepMind, where they reasoned, hey, we're good at reinforcement learning, and we're good at making agents that play games. Why don't we develop a network that plays Folded? And that particular idea didn't progress very far, but the same team kept on working on the problem. And as people became more familiar with the domain, it became clear that there were two important sources of data available to be working with. First and most obviously, we have the collection of all known sequence structure pairs. And back at that time, about 150,000 structures had been painstakingly experimentally determined. So it's a reasonable number, but it's not exactly, big data by the standards of modern machine learning. Very importantly, the scientists working in this field had created a centralized repository for protein structure data called the PDB. These people actually had the foresight to do this all the way back in the 1970s. To this day, the earliest sort of format for storing protein structures was influenced by punch cards, I believe. This was something that took considerable foresight to think of back then. And depositing your results in the PDB was basically made mandatory if you wanted to have a journal publication. And that meant that over the course of 50 years, it grew into this really comprehensive resource. The other major type of data available was multiple sequence alignments, or MSAs. Basically, you can take your target protein sequence and use well-established tools like Hammer or HH-Blitz to search for evolutionarily related sequences in large databases. So here we have a human target protein, and we found evolutionarily related proteins, let's say in fish, rabbit, chicken, etc. And all of those sequences are aligned with each other to make a 2D array. Now, the reason that the MSA is valuable, generally speaking, is that every sequence in it represents a working version of the protein that probably preserves the structure. But there are some differences or mutations between those versions. If two amino acids are close together in the 3D structure, then a mutation in one of them may need to be compensated for by a mutation in its neighbor in order to preserve that 3D structure. And that's going to show up as a kind of pattern in the MSA. So here, whenever residue I changes, residue J has to change as well. That means that an MSA is full of subtle clues about the protein's 3D structure, some of them much more subtle than this. So the first public version of the AlphaFold system basically said, an MSA is a 2D array, and if you squint, it kind of looks like an image. We know how to deal with images. The established off-the-shelf way to process those is with convolutional networks. And so that was essentially the architecture of AlphaFold 1. There is a stack of 220 residual convolution blocks that process features that are based on the MSA. And what was predicted by that network wasn't actually a set of 3D coordinates, but a bunch of much simpler properties that we expect the final protein structure to have. So AlphaFold 1 would predict the distance between each pair of amino acids as a probability distribution over distance bins. It was something that they called a distogram back then, and that's what's shown on this diagram on the right. And it also predicted torsions, which are kind of like the angles that consecutive amino acids make with each other, roughly speaking, as you go along the protein backbone. As for producing an actual 3D structure, well, there wasn't a well-established way of doing that. And so AlphaFold 1 fell back on a non-learned solution and addressed that part of the problem without AI. So essentially, the predictions of the network were treated as constraints on the final protein structure, and they were used to come up with a protein-specific potential. And the team then ran a very expensive search procedure that used gradient descent to try and find a structure that fit all of those constraints. So how well did this approach actually work? Well, in the protein structure prediction world, we had the luxury of already having a single well-established eval that everyone knew to pay attention to, not a luxury that we always have in machine learning. And that was the Critical Assessment of Structure Prediction, or CASP, which still runs every 2 years to this day. And essentially, the organizers ask scientists to volunteer some protein structures that they've just experimentally determined, but which crucially they haven't published yet. So each participating group in CASP over the course of a couple of months is asked to predict these structures, and then a group of experts make really detailed comparisons between the predictions and the ground truth. Obviously, it's a particularly trusted assessment because it's performed on brand new, held out data every single time. And the headline metric in CASP is GDT, which you can think of as roughly the percentage of amino acids that are in the right position when you align the prediction and the true structure on top of each other. So the best possible GDT for a protein structure prediction is 100. And AlphaFold 1 definitely made progress. It was the best performing method in 2018, and it was among early machine learning attempts on this problem that got people excited about the potential of ML in this space. But it was still achieving less than 60 median GDT on the hardest targets, and that limited its practical utility. So the figure above shows an example predicted protein overlaid on an experimental structure from the AlphaFold 1 paper. And while this specific target looks really good, AlphaFold 1 couldn't achieve this level of accuracy consistently. So now let's move into the sort of late 2010s, early 2020s, what I'm going to call the time of specialist models doing specialist things. As machine learning became a much more familiar sight in computational biology, I think we began to see a faster pipeline from the ML literature to the ML for bio literature. Buzzy, popular methods were much more quickly being applied to scientific problems. And at the same time, people seemed to be messing around with network architectures much more and making more extensive changes to specialize them for a particular domain. So rather than applying or just tweaking an off-the-shelf architecture, the goal was now to build in some prior knowledge about the domain and provide good inductive biases for the model from the beginning. And this is also pretty applicable to how AlphaFold 2 was developed. So rather than being a close successor to AlphaFold 1, AlphaFold 2 was actually basically built from scratch, from the ground up. Throw it all out, start again. And it contained a lot more design decisions that were specific to protein folding. Probably the biggest overall change was the move to an end-to-end network. Whereas AlphaFold 1 predicted constraints on a protein structure, but the atomic coordinates themselves were determined separately by a non-learned search process. Within AlphaFold 2, they introduced a structure module, which could produce the atomic coordinates directly. And this creates an end-to-end network that takes in a sequence and outputs a structure. This is beneficial for two reasons. Firstly, sort of from an ideological perspective, the machine learning experts felt that this allows all parts of the network to get a learning signal based on how well the intended final task is performed, not some intermediate subtask like predicting distances or torsions. So in theory, that means that the representations learned from the MSA should now be much better suited to structure prediction. Second, from a very practical perspective, the search part of the old system used to be very slow and expensive. So replacing it with a neural network changed what was actually possible with the model. So for example, we could now consider predicting the structure of all 20,000 or so proteins found in humans, which would previously have been, computationally infeasible. Of course, this meant engaging with the part of the problem that didn't have a clear off-the-shelf solution, how to generate a protein structure. And in the end, the answer was to stop thinking about the protein as a chain defined by a series of angles, and instead think about it as basically a bag of entirely separate amino acids. So each amino acid type contains 3 common backbone atoms, a nitrogen and 2 carbons, that together we can take to define a frame. That's what's represented by the red triangles in this diagram. Knowing where that frame sits essentially specifies the position and orientation of most of the amino acid, ignoring side chains for a minute. So all of the frames in AlphaFold 2 start out at the origin, and the structure module network, its job is basically to predict the rotation and translation needed to move each frame into its final position. Some auxiliary losses do have to be applied to penalize unrealistic output. So for instance, the model producing unrealistic peptide bond lengths, where it's putting 2 neighboring amino acids in the sequence really far apart from each other. But just with that small nudge, the model does eventually learn that proteins should form connected chains, and that aspect of the structure then emerges organically, which is something that kind of makes sense from a computational perspective, but was really counterintuitive for the biology community. The main loss applied in the structure module was also pretty specialized to the problem at hand. Basically, the predicted and ground truth structures are compared many times, each time aligning them on a different amino acid. A clamped L1 loss is then applied, which is based on the atom position errors across all of these different alignments. And because multiple alignments are used rather than one global one, this kind of helps push the model towards a solution that's at least locally correct. There were some equally big changes in how the MSA was processed. The original ConvNet solution from AlphaFold 1 was thrown out and replaced with a transformer-inspired block called evoformer. The purpose of the evoformer block is really to pick up on the subtle clues in the MSA that I mentioned, and then to use that information to iteratively build up a picture of the relationship between amino acid pairs. To make this goal a bit more explicit, a separate pair representation was added in AlphaFold 2, which kind of gives information about amino acid pair relationships an obvious place to live. So within evoformer, the MSA and pair representations can exchange information, and the MSA is first processed through row-wise and column-wise attention, which is a very reasonable thing to do from a biological perspective, since the rows and columns of an MSA do have special significance. Each row is one complete example sequence, and each column corresponds to a particular sequence position and shows how it's varied throughout evolutionary history. Later, the pair representation is updated based on something called triangular attention, in which triples of amino acid pairs are allowed to attend to each other. And this is probably the step that's been most talked about in terms of being based on real physical intuition. In real life, a triple of points in space has to obey certain geometric rules, like satisfying the triangle inequality on distances. So it makes sense to let triples attend to each other in the pair representation, in the hope that the model can then build up a geometrically consistent picture of the relationship between amino acids. That means the model's hypothesis about the structure will hopefully satisfy real-world constraints and be something that can ultimately be realized as a 3D structure by the structure module. So how effective was this new approach to protein folding? Well, AlphaFold 2's median GDT score was around 90, so 30 points higher than AlphaFold 1. And the model was generally much more consistent at producing good predictions, even on the hardest targets, the ones with really low similarity to anything in the PDB training data. And AlphaFold 2 could also scale up to quite large proteins as well, without needing to split them into pieces. That can be really helpful when, you have domains that are packed quite closely together, so the context from the full protein is important to making a good prediction. And you can see that in the example I've chosen here from Cas14, which is actually of an RNA polymerase. I think it had a few thousand amino acids. At the time, it was considered a very large, very challenging target. Today, this would be tiny, but that's the way the field moves. Because the model had reached this 90 GDT level of accuracy, it began to be talked about as competitive with experiment, which to me essentially means that it's a useful tool for scientists to get some immediate structural intuition about a protein, rather than having to remain in the dark until a very time-consuming experiment can be performed. And of course, that statement led to this mad rush to actually get AlphaFold 2 into the hands of scientists where it could do some good. I'm not going to talk about that sort of open sourcing effort in a ton of detail here, but it is worth noting that it involved some new work on the machine learning front. And most of that work, as always seems to happen whenever we want to release something, was focused on confidence metrics. AlphaFold 2, as we used it in CASP, came with just a single confidence metric, something called PLDDT. And all that PLDDT indicates is how confident is the model in the local structure around one specific amino acid. So the prediction on the left has been colored by PLDDT, and essentially it's saying that the blue parts are basically high confidence, and the yellow-orange parts are locally low confidence. However, one of the reviewers on our paper pointed out that it would be really helpful to have a second global confidence metric that tells you how sure AlphaFold is of the sort of larger scale structure. And so the team developed that second metric, predicted aligned error, in fairly short order, and a version of it is still in use in AlphaFold 3 today. In general, I think it's great to be reminded that deploying work publicly comes with this additional responsibility to communicate carefully and to at least try and flag up any model outputs that might be wrong. So ultimately, the effort to make AlphaFold public resulted in 3 artifacts: the open source code, a lightweight sort of server-like interface in Colab for running new predictions, and the AlphaFold database, which was a static collection of pre-computed predictions that was released together with the European Bioinformatics Institute. We're really lucky to have them as partners. They're absolute experts in communicating, comp-bio data. So fantastic to partner with them. And the AlphaFold database started out with predictions for all human proteins, as well as proteins from several other model organisms important to biology. And it's since grown to cover a large portion of all the proteins catalogued in UniProt, which adds up to the total of over 200 million today. So a really interesting point to note, and that I want to emphasize here, is that the increased accuracy of AlphaFold 2 was achieved with essentially the same training data set as AlphaFold 1. We didn't so much as move our date cutoff to include the structures that were released in PDB between 2018 and 2020. In science, it is fairly common that we'll be in this situation where your training data is generated by performing an experiment. And each experiment you perform takes time and money. So we can't just generate more training data through simulation, and nor can we expect loads more of it to be generated by the community if we were to only wait a while. Arguably, all of the tricks in models that have been highly tuned for a very specific scientific problem are kind of a way of trying to do more with less. Find a good enough architecture with the right biases baked in so that the problem can be solved even with the limited training data. And it does seem that all of the little tricks and design decisions made in AlphaFold 2 really did matter to some degree. After CASP, the team ran ablations to try and figure out what was the big idea that mattered the most, so that we could write about it in our paper, of course. And awkwardly, the outcome was that there was no one big idea. Instead, lots of little changes that seemed to make a small contribution individually, cumulatively added up to a much better model. And anecdotally, that was our experience during the development too. There wasn't one day when we suddenly woke up and saw our eval results and said, oh, protein folding solved, that's great. Rather, it was a case of maintaining slow and steady progress for a year or two. Now, of course, since then, the model has been developed further by DeepMind and Isomorphic Labs. First, there was the release of AlphaFold Multimer, which went beyond predicting single protein structures, and instead was specialized to predict protein complexes. So that means collections of 2 or more proteins bound together. As I said, really important for biology. This is kind of where all of the action happens in terms of, determining what proteins do. And more recently, AlphaFold 3 was released, which goes even further. So it aims to predict complexes that contain all the major types of biomolecules, proteins, but also DNA, RNA, ligands, and ions. That's the iteration of the model that's currently available through AlphaFold Server. So a much neater, cleaner server interface than the one that we initially released for speed in Colab. The biggest change in AlphaFold 3 was probably to swap out the structure module for a diffusion module. Essentially, the old approach to generating protein structures was great, but it was really, really highly specialized for proteins. Once you start wanting to support other types of molecule, it becomes very unwieldy to have to figure out how to define frames and violation losses for each molecule type. So instead, we moved to this diffusion model, which works with the raw atom positions directly without having the need for frames. And apart from making it easier to support new molecule types, there are other reasons to think that this is a good fit for the problem. Essentially, complexes have structure on a range of scales. And so the idea is that when denoising a structure with a lot of noise added to it, the model is forced to learn about how the large-scale features of the complex look. So how the different components fit together. Whereas in the low noise regime, it's supposed to learn about the fine details. And perhaps because of this, diffusion models, they naturally learn to produce plausible output. They don't need these extra losses to sort of nudge them towards realistic bond lengths, for example. And overall, I think diffusion is a great example of the comparatively fast pipeline nowadays, from buzzy ML idea to concrete applications in science. Even just in the protein space, which I'm most familiar with, there were several applications of diffusion that you could point to quite early on, even prior to AF3 and RF diffusion. So now we come to today, in the mid-2020s, when there's been another revolution in AI research caused by the rise of a different buzzy idea, large language models. 2022-23 was, of course, a crazy time in the industry, with the release of ChatGPT and subsequently Gemini, Llama, Claude, Grok, DeepSeek, and all of the rest. And I think the level of interest wasn't really brought home to me until I began to see multiple AI stories on the front page of BBC News, and my mom and dad started talking to me about AI on a regular basis. Just looking at the, I escaped most of the craziness of this time by going on maternity leave. Very sensible move. Just looking at the Google Trends plot at the top, you can see the steady growth in searches about LLMs as a topic. And although the scales aren't at all comparable, in the second plot, you can see that LLMs for science is now experiencing a bit of an uptick as well. In general, the people who develop LLMs are really keen for them to show promise in science, because that's one obvious way in which they might be able to benefit humanity through new discoveries. So how has this latest revolution influenced the way that AI for science is done? Well, now I think we see a lot more high-profile papers that involve a large foundation model of some kind. Sometimes that large model actually is an LLM, or the authors describe it as a language model. Other times the architecture is just kind of LLM adjacent, or it might be something completely different, but still with a high parameter count. In any case, the hope is that if you train this foundation model on an ungodly amount of data that's kind of related to your problem, it will learn useful abstractions and absorb useful background knowledge. And then if you're interested in a more specific downstream task, you can either use the foundation model or its embeddings directly, or you can aim to fine-tune on a smaller data set that's more closely related to your application. So compared to AlphaFold 2, this is a very different strategy to try and cope with the limited training data that we often face in science. AlphaFold really tried to adapt the architecture to the problem so as to learn efficiently and squeeze as much value out of the data that we have as possible. Whereas now I guess the idea is to cast the net much wider for training data in the first place, aiming to specialize later on. There are certainly plenty of examples where this foundation model approach seems to work, and if I personally have a gripe with it, it's mostly just that there are so few scientific data sets that are really large enough and uniformly processed enough to really talk about training foundation models. So typically in this space, you're either going to be dealing with sequencing data of some kind or the scientific literature itself. Another upshot of basing your scientific work on very large models is that not everybody's going to have the resources to train them from scratch. In the extreme case of the largest LLMs, there's only going to be a handful of teams executing the big training runs. So there especially, people have had to adapt to new ways of working. I think one strategy that's become pretty common is to specialize in generating really clean data sets and good evals for your particular problem. That's always been a good, honest part of machine learning research. It's the sort of thing that I used to do in AlphaFold. But there's a particular focus on it now. And I think that's because if you can convince the people training the large models to adopt your data and adopt your evals, that's one effective way to have influence and kind of make sure that the end product is good at the tasks that you care about. Another approach, especially with LLMs, is to take an off-the-shelf model and just see how far you can push it towards being good at your task without alteration. Prompt tuning was obviously the original way to do this, but now it's a matter of things like enabling tool use or trying to break the task down into simpler chunks that can be accomplished by a fleet of more specialized agents, so agentic approaches. I'm also a big believer in the power of good engineering, and there's definitely value to be had in just making LLM adoption easier for scientific tasks, whether that's simplifying the ingestion of relevant data or hooking up specialist scientific tools. Finally, there are ways to make progress without access to enormous resources, such as focusing on fine-tuning or trying to prove the value of an idea at smaller scales. So what kind of applications of LLMs are we already seeing in science? It's a big space, so I can't hope to be comprehensive in this talk, but I've picked out one or two illustrative examples to kind of look at here. So one application of interest is getting LLMs really deeply involved in the research process, having them review what's out there and then propose new experiments or new hypotheses for scientists to test. This involves synthesizing information, hopefully from different parts of the literature, maybe being able to make connections between domains, and then some long-term planning and reasoning to kind of see what's a sensible idea. And an example here is the Co-Scientist project, which was developed by Google Research and was announced in a blog post earlier this year. So they came up with a pipeline that, given an input research question, will output a ranked list of hypotheses to follow up on. And that ranking is based not just on the plausibility of the idea, but also on its novelty to try and push the LLM towards new ideas. It's intended to be a human-in-the-loop system, so not a replacement for human scientists, but something that a scientist would then review and decide which ideas they want to take forward. Under the hood, this is an agentic system, so it's a collection of Gemini-based agents coordinated by a supervisor. And the tasks these agents perform include things like idea generation and then modification or evolution, a tournament-based ranking that pits ideas against each other, and deep review, which amongst other things is looking at how reasonable an idea is in the light of what's written in the literature. So I am not on the co-scientist team, but my understanding is that they are still working with trusted testers in the community to try out the system. And they do claim some interesting retrospective results in their blog post. So for example, in the domain of antimicrobial resistance, there's a particular type of mobile genetic element with a clunky name called a CFPICI. And these elements are widely distributed across a whole range of bacterial species. So co-scientist was asked to suggest how that wide distribution might have come about. And the model came up with a hypothesis that involved the CFP-ICIs packaging up their DNA into capsids that then interact with a variety of phage tails, ultimately allowing them to be spread by diverse bacteriophages. That hypothesis had actually already been experimentally confirmed by a group at Imperial, but their paper was still in review at the time, meaning that co-scientists couldn't have just stumbled across it online, and instead it must have independently arrived at the same idea. Another category of application is training language model architectures on domain-specific data, so scientific tokens, if you will, rather than plain text. As an example of this, I've chosen ESM3, which is by a team predominantly at evolutionary scale now, which was published in Science earlier this year. And the explicit goal with ESM3 is to kind of train a foundational language model for proteins. The system they've come up with is multimodal. It's trained on 3 different types of input data: protein sequences, functions, and structures. Now, the sequences and the functions are already essentially text or text-like, and they're relatively easy to imagine how to tokenize, whereas the 3D structures have to pass through an autoencoder, which is specifically designed for 3D protein structures. To emphasize again the scale of this sort of operation, ESM3 is apparently trained on 770 billion total tokens, corresponding to 2.8 billion natural proteins. So, there's maybe not that many domains where you're going to have that scale of data available. The main result they highlight in their paper is the generation of a novel green fluorescent protein. And the way they achieved this was essentially by prompting ESM3, but because it's not a natural text language model, this was using a structural prompt. So the model's given the positions of certain key amino acids from a natural GFP, and it's kind of invited to invent the rest of the surrounding structure, the part in blue. That process was run several times by their team and involved an iterative optimization of the design sequence and structure. So they took a collection of reasonably novel designs to test in the lab, and after synthesizing and testing about 296 well plates of designs or so, they had a protein with comparable fluorescence to natural GFPs, but with only 58% sequence identity to the closest natural protein. And that's something that we often care about in design because you're looking for a diverse set of starting points, basically, if you're going to try and optimize these further towards some goal. Finally, probably the most widely adopted use case of LLMs by scientists today is simply keeping on top of the vast amount of literature out there and summarizing it. This literature review was generated by the Gemini app, but I could equally well have used the deep research feature, some other LLM, or even thrown a few papers into the Notebook LM app and had them summarized as a podcast if I wanted to. Importantly, most of these models now have access to web search or at least some other means of retrieving information from a corpus. And in my experience, that means that citations are much less likely to be invented than in the early days of LLMs, and factuality is greatly improving. As well as the more general purpose LLMs, there's now a thriving ecosystem of tools specifically for science that try to access a paper corpus rather than general websites. And I thought I'd call out the startup Consensus here because I think they have some interesting ideas around the output format. Particularly this consensus meter is what they call it, which aims to show to what extent the literature supports a particular conclusion. That has obvious potential utility for the general public, if it's working well, or perhaps even for medical professionals who are looking to pursue an evidence-based approach. But the particular application that's closest to my heart also involves processing the literature in a slightly different way that I believe could potentially be game-changing, and that's data curation. Work in AI for science still relies on having the right training data available, and when it came to AlphaFold, we were just extremely lucky that not only had scientists done lots of structured determination experiments, they'd also decided to deposit those results in a large database and put them in a clean, machine-readable format. Now, some fields have a proud history of investing in this sort of data curation, and they spent thousands of man-hours assembling and cleaning data from papers, essentially. I've highlighted some specific example databases here, some of the big ones, PDB, Uniprot, and PubChem. But once you stray outside those areas, well-curated data can be much scarcer. In many subject areas, important information is still buried in the original papers or patents as free text or even figures. So naturally, we'd like to leverage LLMs to improve this situation. Plenty of systems today will read tens of papers for you and produce free text summaries, whereas what would be needed here is a system that can read 10,000 papers for you and at least provide a first pass at a database like PDB or Uniprot. The example of the kind of output that we'd be targeting here is taken from a public demo that is a couple of years old now, actually. And in that demo, Gemini processed a large collection of PubMed papers and pulled out a table of genetic variants and their associated diseases for downstream analysis. So it's easy to underestimate the difficulty of this task, given everything that LLMs can already do today. But good data curation is by no means trivial. First, the cost of LLM calls can quickly add up. So we need to think carefully about using the smallest possible model that maintains accuracy without going overboard in order to build something that can potentially scale to an entire field's worth of literature. At that kind of scale, even a low percentage of hallucinations would lead to an appreciable amount of incorrect information in the final product. So building eval sets to measure like hallucination and error rates and then pushing up accuracy on them would also be another concern. It's also essential that scientists have confidence in the final database, and there are various ways that you can bolster that. It's become quite standard to provide a short verbatim quote from the original source to make manual verification of extracted data quicker. But you could also consider developing a model confidence score, a la AlphaFold, to narrow down which data points most need human review. Finally, well-curated data tends to contain less free text and more sort of standard identifiers, controlled vocabularies, and things that make downstream analysis with code easier. And that means mapping the extracted free text to the best matching standard term. In some fields, standardized vocabularies and ontologies won't even exist yet, and so it might be necessary for an LLM to propose those things on the fly. And inevitably, you'll run into edge cases that are challenging even for human curators to unpick. I've spoken to curators who said that they debate these things for hours, exactly what this should be filed under in the major databases. There have been a few instances of this type of system being deployed. So for instance, researchers at the ARC Institute used an agentic system to produce this data set called SC Basecamp. And basically, they took all of the single-cell RNA-seq data in the sequence read archive and tried to curate metadata about where exactly those samples had come from in order to increase the utility of the dataset to scientists. But overall, I still think curation is an underexploited application, something that you hear much less about than free text summarization. All right, so I'd like to finish with a few hot takes about my current research area, which should be a decent jumping off point for discussion. Just thought it would be something nice to do at a summer school. And I understand that this talk is being recorded, so there should be plenty of opportunity to recover it in 10 years and embarrass me with all of the stupid stuff that I said. So first of all, I do believe LLMs have a great deal of potential, but I think we need to be a bit wary of throwing them at every scientific problem, whether that means taking an off-the-shelf model as a starting point or just using an LLM-inspired architecture. Sure, they're a very sensible baseline to run. I can see that everyone ought to check if Gemini or ChatGPT has already solved their problem. But for my money, it is still the case that there should be a reason behind your project's main architectural choice. Some justifications for picking an LLM might be you have input that's naturally text or text-like, or you want to leverage the prior knowledge about the world that a trained large language model has absorbed. Or maybe you need to generate some kind of human-readable output, like a rationale. Although even there, I would caution that models can sometimes make up a plausible-sounding reasoning that isn't very helpful. Second, I still believe that sort of specialized domain-specific models will continue to play an important role in scientific discovery. Right now, if you listen to some commentators, it feels like some people believe the future of science and AI is going to be LLM agents poring over the literature, talking to each other, and then serving up this increasingly inspired list of hypotheses for humans to go away and test in the lab. Essentially a kind of robot scientist that learns by reading. And I think that if you rely only on what's written in the literature, you will never reach superhuman levels of insight. because you're limited to the information that your predecessors chose to communicate in the literature, what they chose to publish, with all of its biases. One example that often comes into my mind is the discovery of this protein with a particularly interesting new function called midnalin. Basically, it was found by looking for things that are involved in protein degradation, but they had to run this sort of large, high-throughput experiment to try and narrow down which proteins might have been involved in that. And they ended up pulling out something that hadn't been written about very much. It was sort of in the dark matter of the human proteome. And I feel like a discovery like that has to be made by either running new experiments or going back to raw data. It isn't something that you would necessarily have been able to infer from the literature. So for me, truly superhuman abilities come from being able to go back to the raw data and derive something novel from it, or from being able to make such accurate predictions that maybe you don't need an experiment in the loop every single time. And for me, those are areas where highly tuned domain-specific models shine, and it's part of the reason why I'm still thinking about how to gather training data for those sorts of models from the literature. So I expect we'll still continue to train such models in science, and that they'll probably turn out to be complementary to LLM systems. I could imagine them being used as tools by agents. I think there are people who've hooked up agents to AlphaFold, for instance, or these networks could be used to embed scientific data in some way. But I don't see the future of science as being vanilla LLMs all the way down. Third, I think, regardless of anything else, the way people use the scientific literature is changing. LLMs are already a really powerful tool for summarization, as we've seen. And so instead of a scientist accessing like a handful of papers for personal use, I think it's increasingly likely that they'll access hundreds of papers and ask an LLM to trawl through them and pull out the relevant data. That way of working is something that's feasible today for open access papers, but it's just not the pattern of access that existing journal systems are built for. If LLMs do become an indispensable tool in processing the literature, I kind of wonder if there's going to be a clamor for new ways to access library holdings too, so that people can access the necessary full texts at scale. I think there's also a bit of a broader issue here, which is that chatbots are making it ever easier for the general public to engage with science, to satisfy their curiosity, and also sometimes in a medical context. So I'm aware that their access is going to be limited to whatever's been made publicly available. Finally, I'd like to emphasize that we kind of need to keep our eyes on the prize. If we're in AI for science specifically, and not just, the field in general, then our prize is surely to advance human knowledge, and by doing that, kind of improve people's quality of life. So working with LLMs is exciting and buzzy and great, but only insofar as it serves that goal. If we look back 10 years from now, and we find that we've built a dozen nice frameworks and some really shiny products, but we've made no progress on issues like room temperature superconductivity or a means to prevent dementia, then we've probably been applying our skills in slightly the wrong places. And I'll end on that challenge. Before I go, I just need to thank some of the people who made the work in this talk possible. On the DeepMind side, firstly, the AlphaFold team. This is the closest thing we have to an AF2 team photo, given that most of our work took place during the pandemic. But many more colleagues, both at DeepMind and at EmblyBI, who I've mentioned, have contributed to open sourcing, to the AlphaFold database, AF3, and the server over the years. Additionally, AlphaFold wouldn't have been possible without decades of effort by the scientists who gathered our training data and deposited it in the protein data bank, and by the scientists who developed the CASP assessment and all of their wonderful evals and metrics. Finally, I'd like to give a special mention to Lindsay Wilmore here, who worked on AlphaFold 3 and is now my main collaborator on the newer LLM stuff, with support from Taylor and Atavia. So happy to take any questions on my hot takes or otherwise. Thank you.